Hi everyone, my name is Mohammed Hassan. I am a PhD student at University of Calgary. Today, I'm going to present to you a study on the pathogenicity of the Canadian DMV 1639 infectious bronchitis virus in layers at different age of exposure to the virus. Infectious bronchitis virus is a gamma coronavirus that belongs to the family Coronavirinae. The virus exists as many different serotypes and genotypes, which complicates the process of the disease control. The disease, known as infectious bronchitis, is a highly contagious disease of chickens with a clinical presentation that varies with the infecting virus strain and age and immune status of the host. The disease is primarily characterized by respiratory manifestations. However, some strains can infect the renal system, causing renal disease, and even fewer strains can cause reproductive disease. Of these strains, M41, Australian T, and QX strains are well known to cause reproductive IB. When infection occurs in young birds, it can cause cystic lesions in the developing oviduct, as shown here in the image on the upper right side. This problem is known as false layer syndrome, and flocks with false layers usually do not reach the production levels of a normal flock, as shown in the lower graph. But when infection occurs in adult birds, it can cause drop in egg production and production of eggs with inferior quality. This graph shows the strains that have been dominant in the Canadian poultry operations between 2015 and 2018 that involved Mass, Con, California 1737, DMV 1639, and 491 strains. Toward the end of 2015, several egg production problems started to be reported in Ontario and Quebec, in which more than 50% of egg layers are located. The strain DMV 1639 was isolated from a number of these flocks, which created an association between the prevalence of this strain and the egg production problems reported. So, we hypothesized that infection of young and adult laying hens with the Canadian DMV 1639 causes gross and microscopic lesions in the reproductive organs that adversely affect the egg production. And the objective of this study was to investigate the pathogenicity of this isolate to the reproductive organs of 1-day-old and adult chickens. In the first experiment, we used 40 specific pathogen-free 1-day-old females that were divided equally into two groups. One group was challenged with the Canadian Delmarva strain at a dose of 10 to the power of 6 imperial infectious dose, and the second group was kept as negative control. Both groups were monitored for disease progression with weekly collection of oropharyngeal and cloacal swabs. At 1 week of age, 5 birds from each group were euthanized, and at 16 weeks of age, all the remaining birds were euthanized. At both time points, portions of different organs with particular attention to their reproductive organs were collected on RNA safe and formalin. Weights of ovaries and lengths of oviducts were measured at the end of the experiment. We used a clinical scoring system that gave a score of 1 for a number of general signs and a score between 1 to 3 for the respiratory signs according to the severity. We started to observe the clinical signs between 3 days post-infection up to 14 days post-infection that peaked at 6 dBi and started to decrease between 8 and 9 dBi. Any bird that received a cumulative score of 5 was euthanized, and birds in the infected group were euthanized between 5 and 12 dBi with a cumulative mortality of around 19%, with no mortality reported in the control group. However, there was no statistical significant difference in the survival rates between both groups. Using qPCR, we quantified the viral genome load in both oropharyngeal and cloacal swabs that showed a continuous virus shedding in up to 21 dBi in the oropharyngeal route until the end of the experiment for the cloacal route. During post-mortem examination at 16 weeks of age, all control birds showed normal, immature reproductive organs as shown here in the upper image, while around 40% of birds in the infected group showed cystic formation with varying sizes that were filled with clear fluid as shown here in the image at the lower part. We used a lesion score for the cystic formation in the oviduct with 4 grades: negative, moderate when cysts are covering less than 20% of the oviduct, marked when cysts are covering between 20 and 50%, and severe when cysts are covering more than 50% of the oviduct. And accordingly, most of the birds in the infected group had marked cystic lesions, with significantly lower healthy birds in the infected group compared to the control group. Microscopically, all parts of oviduct, magnum, isthmus, and uterus of the infected group were affected. They generally showed loss of cilia with a sloughing of epithelium. Areas of edema and mononuclear cell infiltration were also evident, and the marked glandular ectasia was observed in the lamina propria of the part that was affected with the cystic lesion. There were no statistically significant differences in the lengths of oviduct and weights of ovaries between the infected and control groups. In the second experiment, we used 20 specific pathogen-free hens at the age of 26 weeks, and they were at the peak of egg production, and they were divided equally into 2 groups, challenged and control. They were monitored for disease progression and egg production for 10 days post-infection. At both 5 and 10 dBi, 5 birds from each group were euthanized, and oropharyngeal coloacal swabs were collected. Portions from different parts of the oviduct were also collected on RNA-safe and formalin. Percentages of daily egg production were recorded for both groups, and the egg production dropped to 40% in the infected group starting at 5 days post-infection and continued between 20 and 40% till the end of the experiment. During post-mortem examination that was done at 5 days post-infection, there were no obvious gross lesions in the reproductive organs of both infected and control groups. But at 10 days post-infection, 2 birds in the infected group showed ovarian regression and atrophied oviduct, as shown here in the image at the right side, compared to normal reproductive organs in all birds in the control group, as shown here in the image at the left side. The histopathological changes observed at 5 days post-infection were minimal compared to the changes observed at 10 days post-infection. Generally, different parts of the infected oviduct, magnum, isthmus, and uterus showed epithelial sloughing, and areas of focal and diffuse mononuclear cell infiltration were also evident, as shown here in the infected magnum. Atrophied glands with fibrous connective tissue proliferation were also evident, as shown here in the infected isthmus. The infected uterus had areas of edematous and inflammatory cell infiltrations. In conclusion, we found that experimental infection with the Canadian DMV 1639 in young layer chickens does cause cystic lesions in the oviduct, which leads to the false layer syndrome, and in adult laying hens, it induces pathological changes in the reproductive organs that adversely affect egg production. And for future work, we are in the process of analyzing samples collected from an experiment to evaluate the efficacy of the Canadian IB vaccines against the reproductive effects of the Canadian DMV 1639 infection. Finally, I would like to thank my supervisor, Dr. Faisal Karim, my supervisory committee, our collaborators from University of Guelph, my lab members, and my funding agencies, and thank you all for listening. And I'm happy to take any question.
Well, Mohammed, thank you very much for your presentation. This is a very timely piece of research since out east we are experiencing various cases of false layer syndrome in both in Ontario and in Quebec. So thank you for your work. I have no questions at the moment from the audience. However, I have one for you regarding your birds that you infected at a day of age. You did terminate the trial at when they were 16 weeks of age. Would you have or do you have any work where you have infected them at a day of age and kept them up till late?
No, actually, this is the experiment that we've done for now. We infected them at 1 day of age, then euthanized all the birds at 16 weeks of age. Since these false layers at this age or even further, they don't show any clinical signs. That's why we euthanized them at this age to see the lesions of the cystic oviducts.
Yes, indeed. What we've done, we've done some work with ultrasounds in layers at both 18 and 25 weeks of age. And we did show that there's a cystic oviduct initially, but eventually it becomes more difficult to diagnose the cystic oviduct with ultrasounds as many birds are showing atrophies and I'd say incomplete oviduct. They're showing atresia or partial atresia, but the cystic lesions do tend to decrease. So I was wondering if you had done anything in that regard, but unfortunately not. The other question that I have you seen kidney lesions in your infected birds?
You're asking about which age?
No, I'm wondering, have you seen kidney lesions in your birds? Did you look at kidney lesions?
Yes, since we should see these kidney lesions at younger ages, we submitted the grossly, we saw some pale kidneys and swollen kidneys, and histopathologically, we're waiting for the slides. We submitted them, but there are some delays for the histopathological slides. But grossly at younger age, because we euthanized birds at 7 dBi and then at 16 weeks of age. At 7 dBi, yes, we have seen some pale swollen kidneys grossly, because it's a nephropathogenic strain in the first place.
Okay, so at 7 days, but not later?
No, not later.
Okay. Well, I guess that this is all in terms of question. It was very interesting, Mohammed. So thank you very much for your presentation.